I'm Steve Brown, the technical editor of LCGC and Spectroscopy, and I'm here today with Norman Davichi, the Grace Rupley Professor in the Department of Chemistry and Biochemistry at the University of Notre Dame. Professor Davichi, thank you for joining us today.
My pleasure.
Your group was involved in developing a capillary array electrophoresis system with laser-induced fluorescence detection for use in the Human Genome Project. How did that work influence your efforts over the past decade?
That work really stimulated my interest in bioanalysis and big-scale bioanalysis. We had been working on that project for about a decade, starting in 89 and ending right around 2000 when the genome was published. What we have done since then is moved on to look not at DNA and oligonucleotide analysis, but instead for protein analysis and high-sensitivity protein analysis.
Great. In a recent presentation, you discussed the use of a capillary zonal electrophoresis system with electrospray ionization MS detection for proteomics applications. What were the challenges in developing this system, and what are its advantages and disadvantages?
Well, the main challenge, or the initial challenge, was getting a good interface to get analyte from the capillary into the mass spectrometer. And we've been developing fluorescence-based instruments for protein analysis, and it had developed some two-dimensional capillary electrophoresis systems. We modified those systems and took advantage of that interface in order to make an electrospray interface. And this electrospray interface is nice and simple. We simply take a capillary, place it within a glass emitter, and a glass emitter is simply a capillary that's pulled to a fine tip using a pipette puller. Connect these two through a T or a cross and apply voltage to that intersection, and we end up having a very rugged and reliable and remarkably sensitive electrospray interface. That technology was developed in about 2010. We then been exploring capillary electrophoresis with that interface. I should say also that one of the important technologies involved is coupling with a good mass spectrometer, and the new generation of mass spectrometers have some really quite impressive, if not astonishing, performance in terms of sensitivity and mass resolution. And the combination of a nice, stable, robust interface and mass spectrometer has really allowed us to begin to explore capillary electrophoresis as a tool for protein analysis. Advantages and disadvantages. The capillary electrophoresis has two important advantages. One is that it provides extremely high resolution, high efficiency, and fast separations. And the other is that it requires just a minute sample loading. So, for example, getting theoretical plate counts of a couple 100,000 is, for proteins and peptides, for peptides in particular, is pretty straightforward, pretty routine. Loading a few nanoliters worth of sample is, again, pretty routine. But these advantages also are the two disadvantages of capillary electrophoresis. The very fast separations and high efficiency separations really stress the performance of the mass spectrometer in order to keep up with the separation. So peak widths of a couple of seconds are routine. And so the mass spectrometer has to be able to generate parent ion and tandem ion or tandem spectra across that electrophoretic peak in order to capture the peptides that are present within the peak. And similarly, the small injection volumes really stresses the sensitivity of the mass spectrometer and the interface in order to obtain useful information off of the minute amount of sample that's been loaded.
How does that CE-MS approach compare with UHPLC-MS in terms of analysis time and protein and peptide separation?
So that's a question that's a teeny bit too early to answer definitively. We have looked at we've looked at a number of systems now. And one of the issues is learning how to tune the mass spectrometer to work well with the capillary electrophoresis separation. The mass spectrometer manufacturers, of course, tune the instruments to work with typical UPLC type of flow rates. And since we tend to operate at lower flow rates, it requires different operating conditions for the mass spectrometer, and we're beginning to explore those. But be that as it may, what we've discovered to date is that for relatively simple samples, proteomes of moderate to low complexity, the capillary electrophoresis and the UPLC are generating the same number of peptide and protein IDs in the same amount of time, and with a reasonable level of complementarity. As the samples get more complex, the answer gets more complex. So we've looked at, we just published a description of use of capillary electrophoresis for the analysis of the E. coli proteome. And we observe is that for relatively large amounts of sample, the HPLC, UPLC currently is generating about a 50% higher protein and peptide ID rate. However, as you go to smaller sample loadings down to the nanogram, 10 nanograms and 1 nanogram sample loadings, the capillary electrophoresis does much, much better than the UPLC. So for sample limited conditions, the CE definitely wins at this point in time. I think for larger amounts of sample, we'll see. We don't really have a good answer for you yet. The results that the students obtain and present to me are improving at a pretty remarkable rate. We're on a fairly steep learning curve, and it's going to be interesting to see where that learning curve plateaus out.
Interesting. You also discussed a capillary isoelectric focusing technique in your presentation. What are the advantages of that approach for proteomics applications?
As I said, one of the disadvantages of capillary electrophoresis is a small sample loading. And isoelectric focusing allows you to increase the sample loading dramatically, where you can use a microliter or 2 microliter sample loading, which becomes comparable to what you would do in a liquid chromatographic separation. I have to admit, though, that isoelectric focusing requires fairly sophisticated manipulations in order to perform it. And it's a technology that we have explored and have published on. But we're taking a break from it from the moment and instead simply working with zone electrophoresis, which is proving to be much easier to perform. And by doing some stacking experiments, stacking injection conditions, you're able to achieve reasonably high injection volumes with zone electrophoresis. And the performance of the zone and the isoelectric focusing are comparable.
Okay. Your presentation also described a 2D capillary electrophoresis system. How have you used two-dimensional CE in your research?
In the old days when we were doing fluorescence, my goal had been to do the capillary electrophoresis equivalent of a 2D gel on single cells. And so we would fluorescent label proteins, and we perform let us say, an SDS page-like separation in one capillary and an isoelectric focusing-like separation in the orthogonal capillary, and generate a two-dimensional map which gives a reasonable amount of information on the content of that single cell. The problem, of course, is that fluorescence gives you good, wonderful information on the abundance of material, but gives very little information on the identity. So when we shift to mass spectrometry, we rely on the mass spectrometer to provide lots of information. So we've done our 2D separations to date. I guess we've had a couple of papers or a couple of techniques that we've done two-dimensional separations with a mass spectrometer. In one case, we took a mixture of proteins, separated them in one capillary, and at the distal end of that first capillary, immobilized or prepared a microreactor that contained immobilized proteolytic enzyme. The proteolytic enzyme then converted those proteins into peptides that were separated in the second dimension. So you effect a two-dimensional separation first on proteins and then on the resulting proteolytic peptides, and then detect by mass spectrometry. In a second set of papers, we've been using what we call diagonal capillary electrophoresis, which is the same experimental setup as I just described, a capillary with a microreactor at its distal end, followed by a second capillary that will separate the reaction products. But in this diagonal capillary electrophoresis experiment, we have an enzymatic reactor that's going to remove, change the post-translational modifications on the peptides that we introduce into the first capillary. And what we've been looking at are phosphorylation states of peptides. And we have an immobilized microreactor at that first end of that first capillary containing alkaline phosphatase to remove phosphate groups And what we observe are those peptides that don't contain phosphate groups pass through the microreactor unaffected, and those components that do contain phosphate groups will become dephosphorylated, and they'll exhibit a mobility shift, which creates a off-diagonal component in the reconstructed electropharogram. And it has the cool property of unambiguously identifying phosphopeptides within a sample, because only components that have lost phosphate groups are off-diagonal, and alkaline phosphatase only removes phosphate groups. It doesn't affect any other post-translational modifications. So we've been having fun with that, exploring that also.
That sounds like a powerful technique. What do you hope to achieve with your research in the near future?
Our current project that is obsessing us is exploring this technology for the analysis of single cells and the protein content of single cells. We've just completed our first foray into this, and the single cells that we're looking at are embryos from the classic developmental biology model system of Xenopus or Lavus. And what we do is take individual embryos of Xenopus at different stages in development, and I'm working up the data as we speak. So we have cells that are or embryos that have just been fertilized, and then at stages 5, 8, and 11 of development. We use iTrack chemistry to isotopically label the peptides produced by the organism, the proteins produced by the organism at each of these stages of development, and then do, in this case, we're doing capillary liquid chromatography, followed by mass spec analysis. And it's the amount of data that we're able to generate is quite daunting. You have to be careful what you ask for. We have so much data now that we, first of all, spent as much computer time as mass spec time just analyzing the data and extracting out the mass spectra and the relative abundances, but then trying to interpret the data and begin to understand the relationship between different genes as their expression changes during development is really quite impressive, challenging task.
Sounds like an interesting challenge. Professor Davichi, thank you for talking to us today.
My pleasure, Steve.